Good day. My name is Michael Ray. I am science writer for SENS Research Foundation, and I am joined today by Dr. Matthew O'Connor. He is CSO, Scientific Affairs of Cyclarity Therapeutics, and formerly of SENS Research Foundation. We were direct colleagues for many years. Good afternoon, Oki.
Good afternoon, Michael, and I'm actually co-CEO of Cyclarity Therapeutics, but I am indeed the scientific co-founder of Cyclarity Therapeutics.
Thank you for the correction. So what is the big problem you're trying to solve at Cyclarity?
Well, we're targeting oxidized cholesterol, toxic oxidized cholesterols that accumulate in various cells and tissues with age. And broadly, our technology is applicable to many small toxic biomolecules that accumulate in cells and tissues with age. And initially, we're targeting oxidized cholesterol, and our initial disease indications revolve around cardiovascular disease, in which oxidized cholesterol is most implicated.
Good. And talk about the approach that you're taking. It's based on some previous observations with a fairly common chemical, and you've developed your current work from there. So tell us the backstory on where you are.
Yeah, so when I was conceiving of this project, which eventually became a technology and a company, I was looking for alternative approaches to targeting oxidized cholesterols, and look back in the literature for different ways of doing it. And I came across these molecules, which are cyclic carbohydrates called cyclodextrins, that have existed for in chemistry, in industry, and whatnot for 100 years or so. But in the 80s and 90s, there was a lot of work done characterizing the ability of different cyclodextrins to bind cholesterol and different forms of cholesterol. And so some of that proof of concept work was already old news, but I found that it wasn't really being applied much yet to human drugs, but I thought there was some potential there, so started a project on it.
There was one human application, though, that was in use at that time, right?
Yeah, well, actually, there was two. One that is the closest one to what we're working on is Niemann-Pick type C disease, which is a rare orphan disease, a genetic lysosomal storage disease that is actually a defect in cholesterol transport, and those cells and the patients actually hyperaccumulate oxidized cholesterol in their cells because of the lysosomal defect. So that is actually a pretty close analogy, and a lot of what inspired this project was the whole story of how it was first tested in mice in the early 2000s, and then cats that naturally get Niemann-Pick type C disease, and now it's in clinical trials in people with Niemann-Pick type C.
Right. So based on that research that you had seen, you decided that the thing to do would be develop a improved version of that molecule that would be more specific for oxidized cholesterol. So tell us about how you started thinking about that problem and how you proceeded from there.
Yeah, so what we did is we started looking at that cyclodextrin that was already being tested, that showed a lot of promise, but it's not very potent, and so it has to be used in extraordinarily high doses. Whether you're working in cells or in animals or in people, in such high doses that some of the infusions that happen in the patients take 6 hours just to infuse the drug into the veins, the so-called drug, into the veins of the patient. So it's so low potency that it's kind of debatable about whether or not it should even be called a drug. So we went about looking at chemically how it behaved and how other cyclodextrins behaved around cholesterol and oxidized cholesterol. And we came up with an idea that that allowed us to alter the molecule in a way that increased its potency by something like 1,000-fold against the target, which now it makes it much more potent and feasible to use.
Right, right. The other problem with the old molecule was that it was a lot less specific for oxidized cholesterol, and that caused some of the toxicity, right?
Yeah, the generic one that is being used in the clinical trials isn't too bad in terms of specificity. It's just super weak. But yes, we re-engineered the drug that we made to select for the oxidized cholesterol and against cholesterol, and that actually helps us greatly with the safety, because all of the side effects, all of the toxic effects of cyclodextrins that we observe come from its cholesterol binding properties, come from pulling too much cholesterol out of cell membranes and lysing the cells, destroying the cells. And that can cause damage in many parts of the body. And it turns out, especially in the ear, so it can cause hearing loss if you remove too much cholesterol from the hair cells of the cochlea.
Right, right. Some of our listeners may not know, but cholesterol is not just a component of LDL cholesterol that kills us. It is actually a biologically essential molecule, and the main thing that it's essential for is the formation and maintenance of the fluidity of cell membranes. And the cell can't properly move things in and out of itself and carry out other functions if it doesn't have the right amount of cholesterol. And so the old molecule had a tendency at the doses that were being used to actually pull it out of cell membranes, and that was a real problem. Now, on the subject of membranes, contrast the small molecule approach, in your case, genuinely small molecule approach, that you're using with the classic LysoSENS approach that was originally envisioned by Dr. DeGray and that we're still using with some of our other projects.
Yeah. So we're engineering using rational drug design approaches supported by computational chemistry modeling to develop new structures of these molecules. But enzyme approaches are another approach that we were working on previously at SENS Foundation, and they hold great promise. But I wanted to try something different that I thought might be easier and avoid some potential regulatory pitfalls, so that we could get something to clinic and into people perhaps faster than we could with an enzymatic approach. But the enzymatic approach in principle is potentially very good. You can make enzymes theoretically that are very specific. We had a hard time at the time actually making enzymes that were very specific for oxidized cholesterol over regular cholesterol. But in principle, that's not a problem for enzymes, that you can make them exquisitely specific. So it's a cool approach, but one that's fraught with many challenges that haven't been solved yet.
Right, right. You alluded to this just a second ago, but maybe dive into this a bit more. There are actually some regulatory advantages to working with cyclodextrins.
Yeah, and that's a big part of what drew me to them, is that there's a lot of precedent to working with cyclodextrins. For one thing, there is one approved cyclodextrin-based drug. It's called Bridion, and it's for a totally different purpose. It's basically an antidote against a paralytic that's used in surgery. But a different cyclodextrin has gone through the regulatory process before and been approved as a brand new version that was engineered from scratch, and went all the way through the process, went through all the safety testing. So that path has been tread in one way. In another way, it's been tread because other cyclodextrins are used as carriers, as delivery molecules for other drugs, and those are called excipients. And in fact, the cyclodextrin that you were referring to before is used for that purpose already in many drugs. Perhaps hundreds of drugs already contain it, and it's such a safe molecule that it has this regulatory status, which basically says you can mix it into any drug formulation, and it's considered a safe and active ingredient. And so that's just to say that molecules like this are very familiar to the regulators, and in fact, they can be made to be so safe that they can achieve this status that says that you don't have to really test them as new molecules. Now, our drug is a brand new molecule that is going through the whole safety testing process, but it's not something so new that the regulators are saying, oh, we've never seen this before. There's a really high bar for this. You have to look in every nook and cranny for anything that could be weird or unique about this new type of matter. It's not a foreign type of matter to them. It's something they're familiar with. And so it looks like, so far, knock on wood, the regulatory process for us is going quite smoothly.
Great. And so more broadly, talk about the progress that you have made with the molecule, with testing, with where you're at.
Yeah, so we've done everything from on the scientific proof of concept front to the safety and regulatory front to the manufacturing front is all really at an advanced stage now. It's less than 3 years since we launched the company at the end of 2019, and we have already had 4 regulatory meetings in 2 different countries and been admitted to a accelerated regulatory program in the United Kingdom, which we can get to in a minute. But we've been able to show that we can poison various cell types, including macrophages, with oxidized cholesterol, cause them to die or to turn into foam cells, which are essential to atherosclerotic plaque formation, and that we can treat with our drug and show that the foaminess of the macrophage can go away, that the activity of the macrophage can return. We've published some of this work, not yet all of it. We've also been able to scale up the manufacturing of our drug to large scale kilogram scale manufacturing. And in fact, we've just started the GMP quality manufacturing, the first batch of GMP quality drug product, which if everything goes smoothly, will start going into patients middle or late next year. So
just explain to some of our listeners what it means to manufacture a drug in GMP quality as opposed to anything else.
Yeah, so GMP stands for good manufacturing process, and it sounds simple, but it's actually usually takes years to devise a very careful process by which you produce at large scale in an economically feasible way a drug product which is extremely pure and extremely safe and free of any toxic byproducts or side products and such like that. So we have indeed spent several years working on our process. first scaling it up from milligram quantities to gram quantities, and now to kilogram scale quantities, and then tested it for every conceivable impurity, and now we have a process, and it's being made. So it's the kind of thing that you don't hear a lot about. It's not the most exciting thing to talk about, but it's actually the kind of thing that drug companies like ours end up spending an inordinate amount of time and money on getting right.
Yeah, and unfortunately, another SENS Research Foundation spinout called LysoClear ultimately foundered on that very problem. They couldn't get GMP quality of the enzyme that they were producing that, at least in mouse models, looked really promising against a different target. Speaking of mouse models, if my memory is not drastically incorrect, you do have some mouse data with your molecule. Are you able to talk about that?
Well, we don't have good efficacy data to talk about yet. We're working on that. The problem is that there's not good mouse models of atherosclerosis out there. They don't really reflect what happens in human disease, but we have excellent safety data in mice and rats. So we can give our drug to rodents in extraordinarily high quantities and show that it's extremely safe. We can look at the pharmacology of how the drug behaves, and show that it's not metabolized, that it's excreted, and it's not accumulating in the animals. So everything about the pharmacology and the safety of the drug is looking extremely good. So we're doing those GLP safety tests, good laboratory practice tests, which are required by the regulators before you can start human clinical trials. We're doing those now, but we've already done non-GLP versions of them. So we've got a pretty good idea of what the results are going to say.
Right, great. And so you've set me up very nicely here. All of this stuff is leading directly to the fact that you're coming up very close to the point where you can actually do human clinical trials. And this has happened at a very fast pace. How are you able to do that?
How are we able to move so fast?
Yeah.
Well, it was part of the grand plan to start with, and somehow it's actually worked out that way. And a lot of it was due to the nature of what we talked about before, that we're working with molecules that we had a good plan for how to get them made safely and be something that the regulators would be comfortable with. And the other part of it is the amazing team that we've been able to assemble, and who work extraordinarily hard, but also smart. So we hired veterans of industry for the different applications, the enabling work for getting into the trials. We hired great biologists who have been down that road before, chemists who know how to do the kind of manufacturing that we need to do, regulatory advisors, and now most recently we're building our clinical trial team, which is now being headed up by our newest hire, Noah Rosenberg, who's a veteran of many drug development processes, and so he's heading up our clinical trial team.
Yeah, and I can certainly speak myself to back when you guys were still part of SENS Research Foundation. Several of the people that were on your staff then, most of which are still on your staff now, were just crackerjack smart and very productive scientists. So that certainly helps for sure. Okay,
no doubt, we would not have been able to get as far as we have, or as quickly as we have without them. It's an amazing team.
Yeah. So how are you going to structure the clinical trial? How's it going to be run? What do you expect the timelines to be? All that stuff.
Well, nothing's set in stone yet,
Mhm.
but we'll have the plan right now is to start perhaps as early as summer of 2023, phase one safety trial in human volunteers who will be dosed first with a extraordinarily low dose of our drug, and then ascending the dose into a level that we think will be very safe, and probably ascending all the way to a dose that will be very safe, but probably beyond what we will need to dose patients with eventually. So we have a single ascending and multiple ascending dose, meaning patients that will get only one dose of the drug, and other patients who will get multiple doses of the drug over a series of weeks. And so that'll commence this summer. And we may or may not be able to include some middle-aged volunteers who would not yet be diagnosed with cardiovascular disease, but who could already have some aging of their arteries, in which we might be able to see some effect of the drug anecdotally, without really powering the studies to show a real efficacy effect. We hope to be able to try it on some sub disease diagnosed normal aging people.
Okay, so that is the difference between using an enzyme and using a small molecule. I was asking a more specific question, I guess, which is the difference between the degradation approach that is the original LysoSENS vision versus the way that you expect the cyclodextrins will work in rehabilitating the macrophage. So can you talk about that?
Yeah, so that is a fundamental difference between the two approaches. So in our approach, the target, the oxidized cholesterol, won't be modified by our drug. It will be mobilized, it will be liberated, it will be pulled out of the cells and tissues that our drug gets to, and that's how it will rejuvenate the cells and tissues that our drug comes into contact with, as opposed to breaking down the target, which is what you might do with enzymatic approaches. So that is a fundamental difference with how our technology works. And so, but either one, I think, could work just fine. I'm not sure that there's in that sense a particular advantage to moving out the toxic cholesterol forms versus degrading them, but that's just a difference in the way that it will work.
Right, right. And as a follow-on question to that, since you are actually like moving it out rather than degrading it right there in situ, a naive person might ask, well, then why isn't that just going to cause more problems? You take oxidized cholesterol out of one place and you stick it in another. Like, what's happening with the oxidized cholesterol?
Yeah, so we think it's getting, and we're still working on proving this, but we think it'll just get excreted. There's two ways in which this is fine. So if the drug holds on tightly onto a molecule and then the drug gets excreted, then that's fine. It just gets excreted. If the drug releases it and the oxidized cholesterol gets excreted, that's fine. If the one place in your body, the one organ that actually has some ability to metabolize oxidized forms of cholesterol is in the liver, where the hepatocytes can, to a degree, metabolize oxidized cholesterol. And this is part of why it's okay to eat oxidized cholesterol, but that you don't want it getting other cells and tissues in your body. So you don't want it accumulating in your macrophages, in your arteries, in your arterial walls, where you don't have enzymes that can either break it down or tag it for transport the way you can with regular cholesterol. So the liver just has an innate ability to handle that. So when you mobilize it and you get it into the bloodstream, it actually ends up being much less dangerous. It's just the body hasn't evolved a natural way to do that. So we created that with our drug.
This is fantastic. I mean, you've moved very rapidly, as we've discussed, and getting very close to what is still, the number one sort of isolated disease of aging killer in developed countries and really globally right now. So good luck and Godspeed.
Thanks so much, Michael. It's a privilege to get to work on this every day, and it's been a lot of fun to come back and talk to you about our work and our progress.
Hear, hear.